time to level up and discuss levels of protein structure. So there are 4 levels of protein structure that lead to the prettiness of proteins, and also how those proteins are able to function. These are the primary structure, which is just the sequence of amino acids, so which individual protein letters are linked together in what order. Now, once they link together, they start to fold up. This folding involves secondary structure, in which the generic parts of those individual amino acids, so like the backbone of those amino acids, the part that allows them to link together, it also allows them to kind of like interact with other amino acids that are not directly next to them. And so we call this the secondary structure, where we have interactions between the backbone of different amino acid residues. So basically, a residue is when these amino acids link up, and the leftovers is a residue. And so when the backbones of these interact, this leads to secondary structure, things like alpha helices and beta strands and pleated sheets. Now, you can also then get tertiary structure, in which the side chains, or the unique parts of those individual amino acids, start contributing to the structure by making interactions with one another. And then finally, we have quaternary structure, which is where you actually have multiple protein chains involved. So proteins can be made up of multiple chains of these amino acids, and we get multiples and things like this. And in this case, we'd have quaternary structure, in which these different chains are making interactions with one another. Some of these interactions lead to the distinct shapes of different proteins, and these shapes contribute to how these proteins function. And so there's a field of biology called structural biology that actually tries to deal with trying to figure out what different proteins look like and how their shapes kind of match their function, and their function kind of is made possible by their shapes. And so they use techniques like X-ray crystallography and cryo-EM and NMR and stuff to try to get information about these shapes. Speaking of shapes, proteins can have different regions with different shapes, and so we call different regions of proteins domains. It's kind of like rooms in your house. And then within those individual domains, you can have motifs, which are kind of like characteristic structures or common folds you might find in multiple different proteins, kind of like furniture in the rooms of your house. And so there's also regions of proteins that don't have a defined structure. We can call these intrinsically disordered regions, and these can actually serve really important roles because they can help bring proteins together and allow proteins to like respond to different things. So there's a lot of really cool stuff we can talk about when it comes to protein structure, and at its heart is those 4 main levels of structure. So let's go into those. Remember, this is the primary structure, the sequence of amino acids, the secondary structure, in which we get the backbone coming into play. We get like things like alpha helices and beta strands. This is involving hydrogen bonding between the backbone that we'll talk about. Then we get the tertiary structure, which involves the side chains, and finally quaternary structure, which not all proteins have, but some proteins do, and this involves interactions between multiple chains of amino acids. So let's now talk in more detail. All those fancy structures of proteins start with how they are composed of individual amino acids, and what order those individual amino acids are in is going to help dictate how the protein is going to fold. So there are 20 main genetically encoded proteinogenic amino acids, so basically 20 main amino acids that can be included in the instructions for making a protein and then incorporated into the protein. And there's also things like post-translational modifications, where these amino acids can be altered after they're added to a protein. But these are the 20 main amino acids that are added to a protein as is, like this, and then they can get modified later. But even without being modified, you can see that there's a great amount of diversity of these different amino acids. So each of these has the same generic backbone, which has this amino group and this carboxyl group, and those linked together. The amino acids link together, sharing those by joining up those generic parts through these like peptide bonds. And then each of them also has a unique part or side chain or R group that sticks off, kind of like a charm from a charm bracelet. And this is where the amino acids really vary. And these different amino acids have different side chains with very different properties.
These include things like great differences in size as well as hydrophobicity. So basically whether or not water will hang out with them. If water will hang out with them, then these amino acids can be found on the surface of proteins. But if the water doesn't want to hang out with them, then they're going to be kind of like forced to huddle together in the center of a protein, assuming we're talking about like a water soluble protein. Some of these amino acids most of them are neutral, but some of them can be charged. We can talk about basic and acidic amino acids, and we'll get into this. But basically, you have all these different qualities of these amino acids, and the protein, when it's folding up, it has to accommodate all of those different desires. So, does it want to hang out with the water? Does it want to be near something positive or negatively charged? Does it need extra room? Is it really big and bulky? Does it have to worry about steric hindrance? So basically, atoms need their elbow room. And so if there's a big bulky amino acid, it's going to make it harder to rotate, things like this. And so all of this is going to influence how the protein folds up. And it's going to influence things at the level of the secondary structure and the tertiary structure and the quaternary structure. So the primary structure is really of primary importance. And this primary structure is just what we call like the order of amino acids. And so this is something that you could see even without seeing like a folded protein, just by looking at a sequence, you'd be able to tell the primary structure because the primary structure is just the sequence of amino acids. So those individual protein letters, different proteins can have different orders of these letters as well as different like combinations. Some proteins have a lot of letters and some don't have that many. So we can get big differences in protein sizes as well. So all of that is the primary structure. And the primary structure is going to influence the other levels of structure, starting with the secondary structure. So in the secondary structure, although we're just talking about interactions involving the backbone, which is the generic part of the amino acids, the primary structure is still going to come into play because it's going to influence the kind of like how that backbone can rotate and therefore what interactions it can form. So when these amino acids link together, they do so by making these peptide bonds. So in a peptide bond, you have the amino group of one amino acid combined with the carboxyl group of another amino acid, and this forms this amide bond, which we call a peptide bond. Now, much more in other posts, but this peptide bond has special qualities. It has this like partial double bond character. Um, Basically, you get these electron delocalization, much more on this in other posts. But basically, when you see one of these bonds, one of these lines, it's representing these atoms sharing pairs of electrons, which are smaller, like subatomic particles that make up these different atoms. And by sharing these pairs of electrons, they're able to form bonds. And if they share a single pair, they make a single bond. And if they share two pairs, they make a double bond. And sometimes they do this special thing where they kind of have more than enough for a single bond, but not enough for a double bond. So they kind of share their like quote unquote extra electrons with multiple atoms, and we get this electron delocalization or conjugation or resonance. And for reasons outside the scope of this post, you have to have this happen in a plane. And so these amino acids, they have this resonance in this peptide bonds. You have electron sharing between this oxygen and this nitrogen and this carbon. So this has to be planar. You can't rotate in between these, you can only rotate around them. So at the end of the day, what you end up with is you have kind of like a chain of planes where there's only specific places in the backbone of the protein that you're able to rotate around in between those individual peptide bonds, but not within those peptide bonds. So there's distinct angles that these amino acids that the backbone can take, and those backbone angles are going to be further restricted by the characteristics of the amino acid side chain. If you have something really small and flexible, you're going to have like a glycine where it only has a hydrogen as its side chain. You're going to have a lot of flexibility. You're going to be really loosey-goosey. If you have something big and bulky, you're going to have to worry about that steric hindrance, that atoms saying, get out of my space. And it's going to be harder to rotate. And finally, if you have proline, well, this is the most like structurally restricted because its side chain actually kind of loops back on itself. And as we'll see, it kind of then like steals one of the hydrogens. So you're not going to be able to contribute to the secondary structure. So the individual amino acids, the unique side chains are going to influence the angles, but there's characteristic angles that these amino acids can take on. And when these amino acids, when the backbone takes on those different angles, we call these dihedral angles because they're angles between planes. Basically what can happen is you're going to have these layouts where you're going to have the backbone oriented in similar ways in different proteins, at least in structures of these proteins. And when those amino acids are kind of, when the backbone is oriented in that way, you're going to be left with distinct patterns of the hydrogens and the oxygens in that backbone kind of being available for what we call hydrogen bonding. So remember, in the backbone, we have the generic parts and the unique parts. And the generic parts are going to involve this peptide, in this peptide bond, this amide bond, this nitrogen and this oxygen, the night and the carbon, but it's not going to be involved here, indirectly at least. So this nitrogen is going to be attached to a proton, so to a hydrogen. And then this oxygen is going to have a lone pairs of electrons, so unpaired electrons. And why this matters is because this is going to allow for something called hydrogen bonding. So remember how I said that all these atoms were forming bonds by sharing pairs of electrons? Well, electrons are negatively charged. And so if those electrons are not shared evenly, you're going to get a distribution of charge. And it turns out that oxygen and nitrogen don't share fairly. They're what we call electronegative. So if you think about these electrons that they're sharing as kind of dogs, and the atoms, like the protons, so the central part of the nucleus is positively charged and it's kind of like raining in those dogs. You can think of the oxygen and the nitrogen as kind of pulling harder on those shared electrons, pulling tighter on the leashes of those dogs. So they're able to attract the electrons, not just of their own, not just the ones that they like started with, but also the ones that they're sharing. And this is going to lead to a partial separation of charge, something we call polarity. And when you have charges, even if they're partial charges, even if they're temporary charges, temporary full, whatever, these can form attractions and repulsions. So opposite charges are going to attract one another. And this is the basis for how we can get these sort of like dipole attractions. So a dipole is where you have that separation of charge, and then that separation of charge, those charged regions can be partially charged regions can be attracted to one another. And this is what we see in a hydrogen bond. And so a hydrogen bond is just a special instance of these like dipole interactions, where one of the attracting partners, the partly positive part, is going to come from a hydrogen that's attached to an electronegative atom, something like a nitrogen or a hydrogen. That nitrogen or that hydrogen is going to pull on the shared electrons, making that hydrogen partly positive, and thus able to serve as a hydrogen bond donor. Now it needs an acceptor, and that acceptor is going to come from a lone pair of electrons on an electronegative atom, such as an oxygen or a nitrogen. And so if we think about a peptide backbone, well, we have both of these things happening in the backbone. We have the hydrogen on the nitrogen that can act as a donor, and we have the lone pairs and the oxygen that can act as an acceptor. And when we do this, we can see, I just wanted to mention too, that although it looks like nitrogen has a lone pair that can be involved in this, what's actually going to happen is that lone pair is involved in the resonance. And so you're not going to see that this can't be available as an acceptor, but it can be available as a donor because of that hydrogen. So you see this nitrogen acting as a donor and the oxygen acting as an acceptor. And now you think about the backbone of the protein, it's going to have this like regular pattern of acceptor, donor, acceptor, donor, acceptor, donor, acceptor, donor. And we talked about there's only distinct places where that backbone can rotate. So this is going to lead to distinct patterns of structure that you can find in many different proteins. The core protein secondary structure, or motifs, So these kind of like patterns. We can talk about alpha helices, which are kind of like these helical structures. And these are going to involve hydrogen bonding between the backbone oxygens and the backbone nitrogens for residues later. Then we can talk about beta strands, which are these individual strands. And these can then team up into beta pleated sheets. So these kind of pleated, they're kind of like an accordion style. And these can be parallel, where the strands are running in the same direction, or anti-parallel, where they're running in opposite directions. And so these are the core secondary structural motifs, and they have common representations where we'll see them as kind of like these flat arrows for the sheets, for the strands. And these ribbon helices structures for the helices. And we have Jane Richardson to thank for this ribbon diagram representation. And she's really, really awesome. And I've gotten to meet her a few times at the Cold Spring Harbor Laboratories X-ray crystallography course, which I got to sit in on a couple times. And it was really awesome meeting her, and she's really cool. And she came up with this representation for the protein structures. So those are the secondary structural motifs, where you have the basic structural motifs, where you have like alpha helices and beta strands and beta sheets. Now, those protein core secondary structures can join together to form super secondary motifs. So super above, so these are more than just a single secondary motif, where you can kind of have like strands and helices coming together and things like this. So an example would be a zinc finger, or a beta turn, or a helix turn helix, a beta hairpin, a Walker ATP binding motifs. So sometimes these motifs have a known function. For example, zinc fingers typically bind to DNA, but they don't always have a known function. Another thing is going to be that they can be part of a larger domain. So a domain, this is kind of like a room in your house, and then the motif would be kind of like a furniture in that room. And we'll talk more about that later. But for example, you might have a zinc finger motif in a protein's DNA binding domain. And in fact, zinc fingers are a main way, or a key way in which that allows proteins to bind to different DNA. And because you have multiple proteins that are going to be needed, that need to bind to DNA, you'll find the zinc finger in multiple different proteins. And so these super secondary motifs can often be found in a variety of different proteins. It's not restricted to being in a single protein, but different proteins will have different like combinations of these super secondary motifs, as well as other things. And when we get into all of this and this variation between these proteins, well, now, even though all these proteins have, basically, most proteins have alpha helices and beta strands and beta sheets, they'll have different combinations, like different proportions. So you can have some that are going to be more helical, some that are more like strandy. But you can basically, all these different proteins are going to have these different combinations, but these core motifs are going to allow them to kind of have that starting structure to kind of build off of. And when they build off of it, they're going to often now we get involved with the tertiary structure. And so the tertiary structure, now we're going to directly involve the side chain. So we've been indirectly involving the side chain. So in the secondary structure, remember, the side chain was kind of going to influence the angles that the backbone could take on due to things like steric hindrance, so those amino acids needing their space. And also in the special case of proline, although it looks like it has a hydrogen that can be acting as a donor, remember that you're going to have a lone pair of electrons to contribute to the resonance of the peptide bond. And so once it links together into a peptide bond, it's no longer going to have a proton available to donate. And therefore, if it doesn't have a proton to donate, it can't act as a hydrogen bond donor. And so it's not going to be able to contribute to this, to the secondary structure, at least to those hydrogen bonding interactions. And then with glycine, you're going to have a lot more flexibility. It's going to be harder to form distinct structures. So you typically wouldn't find those in like an alpha helix or a beta strand. And then for some of these, they're like too big and bulky to be found easily in alpha helix, and you're more likely to find them as strand and things like this. So the backbone, so the side chain was influencing the secondary structure, but not directly. It wasn't directly involved in the hydrogen bonding interactions that are giving you those secondary structure motifs. But in the tertiary structure, now they're going to directly come into play, because in the tertiary structure, we're going to get interactions that directly involve the side chain. So the side chains can make interactions with one another, and they can also make interactions with the backbone and things like this. Now, different amino acids have their unique side chains, have different like desires and abilities to form interactions. And when I talk about these interactions, they're mainly going to be non-covalent interactions. So these are going to be attractions and not actual bonds. The one exception that we'll talk about is in the case of cysteine, where cysteine can actually form disulfide crosslinks. So it can actually form covalent bonds. For reasons I talk about in my cysteine post, basically bonds to sulfur are going to be weaker. And so although cysteine can form these disulfide bonds, these like crosslinks, these are going to be able to make, and you're going to be able to break them. And so when they form these crosslinks, we say it's been like oxidized. And then when these are broken up, it's reduced, and much more on this in my post on cysteine. But basically for this, just know that cysteine is able to form these covalent linkages. This can be within a protein chain, so that would be tertiary structure, or between chains, which will contribute to the quaternary structure. But this is basically the only instance where we're going to see that we actually have this covalent bonding of the side chain involved in the tertiary structure. And typically what we're dealing with is really just partial charge, or full charge attractions. And so some of these amino acids are going to be what we call polar. And when you have a polar molecule, basically that's where you get that uneven sharing of electrons. And so carbon and hydrogen are going to share pretty fairly. So if you have something that's purely a hydrocarbon, like you get something that's non-polar, and these non-polar molecules are going to have pretty fair sharing. But then some of these molecules have nitrogens and oxygens, which, as we talked about, were electronegative. So these are going to share unfairly, lead to polarity. And when you have polarity, you have that separation of charge, you have that partial positive, partial negative regions. And this allows for opportunities for these charge-based attractions. In some of these amino acids, you actually have full charges. So these are the basic amino acids, histidine, lysine, and arginine, which are sometimes positively charged. Most of the time, they're positively charged in the case of lysine and arginine, whereas histidine, you can find it in significant amounts of both charged and neutral. In terms of negatively charged, we have aspartic acid in its negative form. Its conjugate base is aspartate. So basically, if it gives up a proton, which it normally does, you'll find it in this aspartate state, and then you have glutamate. And so these will be negatively charged. When we have a fully charged molecule, we call it an ion. And when you have a positive and a negative ion kind of hang out together, what you get is a neutral combo called a salt. And so when we have an interaction between one of these positively charged side chains like lysine, arginine, or histidine, and one of these negatively charged side chains like glutamate or aspartate, we call this attraction an ionic bond or a salt bridge. And so although we say like ionic bond, it's really just a strong attraction. They're not actually sharing electrons. So this is not a covalent bond. It's not a shared pair of electrons. We don't draw an actual line between them, but it's an ionic quote unquote bond. It's this strong attraction. And so you can get this between the charged amino acids. And then you can also get between those other amino acids that aren't charged, you can still get these dipole-dipole interactions and things like this. But actually, one of the key driving forces of the folding of proteins is going to be the interactions that are actually not through these charge or even partial charge, or at least not permanent partial charge. So when we talked about those polar amino acids, we were talking about them having a permanent dipole. So in those electrons that are shared, they're going to be moving around randomly, but we can kind of predict where they're more likely to hang out. So in a polar molecule, they're more likely to hang out around the more electronegative atom. And this leads to that permanent dipole. If you have a nonpolar molecule, well, then the electrons are kind of just going to be randomly moving around, and you're going to have an equal probability of finding them anywhere. But what happens is that if something charged comes near, this can induce a dipole, where basically if you have something negatively charged come nearby, the electrons are kind of going to be repelled from it, and they're going to shift. And this is going to lead the region that they left partly positive, and the region they go to partly negative. You'll have the opposite thing happen if you have something positive appear. You're going to have the electrons kind of go towards the positive, and therefore you're going to get a shift in the opposite direction. So you can get these induced dipoles. And even if you don't have a charge nearby, you can end up with kind of like a random instantaneous dipole, something we call like a dispersion force, a London force, a van der Waals force. There's different names for it. But basically, because those electrons are moving randomly about, if some of them just happen to randomly kind of clump up in one region for even temporarily, this is enough to set off a chain reaction between these different amino acids. And so you can get these non-polar interactions, these van der Waals forces and things like this, that are going to allow these side chains, even the non-polar side chains, to contribute to the structure of a protein. And all this is going to be promoted by the fact that those nonpolar amino acids are going to be excluded by the water. You're going to get the hydrophobic effect, where the water molecules are going to try to maximize their water-water interactions, minimize their interactions with these nonpolar things. You get these nonpolar things huddled together in like the center of a protein. In the case of a water-soluble, or like a cytosolic protein or a excreted protein, basically anything that's not membrane bound is going to be surrounded by water. And so those hydrophobic amino acids, the ones that are excluded by water, are going to be found in the center of a protein. And when they're all together, then they're able to kind of sync up their electrons through these like chain reactions and things like this, and you get these hydrophobic interactions. Okay, so tertiary structure. We have the partial charge base attractions, which are based on, we can have these, the polar molecules are able to always do that, like permanently be polar. And then we have the nonpolar molecules, which we can get induced dipoles and things like this. So lots of ways that these side chains can interact with one another. And then we can actually have these side chains as well as the backbone interacting with the other, with like other chains. So now we're not talking about the side chain, we're talking about actual chains of amino acids, so actual polypeptides. So the instructions for making each protein are going to be, or each chain of protein is going to be encoded for by its DNA gene. Then you get a messenger RNA copy of that, which gets translated, it's turned into a protein. The ribosome, this protein making machinery, is going to travel along the messenger RNA and piece together the amino acids that are dictated by the messenger RNA. And so each of these basically strands of the mRNA is going to tell the ribosome to make a single strand of protein, a polypeptide. And each of these chains, a single chain might be enough for a full protein, but sometimes proteins are made up of multiple chains. So they have like multiple subunits. And if they have multiple chains, then those chains have to interact with one another to form the whole like complex. And those interactions are going to involve different, so the chain-chain interactions, this is what we call the quaternary structure. So in a protein where you only have a single chain, it would be like a monomer. And so a monomer would not have a quaternary structure. But once you get to oligomers and multimers, then you would. So oligo means few. So a oligomer would have just like a few subunits. A multimer can have like many. And we can classify them based on how many chains they contain. So a dimer has 2 chains, a trimer has 3 chains, and a tetramer has 4 chains. Then you get things like pentamer, we get 5, hexamer, 6, etc. We can further classify them based on whether the chains are the same or whether they're different. So a homomeric protein would have multiple copies of the same polypeptide. So multiple copies of the same chain, such as like a homodimer or a homotrimer. A heteromeric protein would contain at least one polypeptide that's different than the others. So you would get a heterodimer, heterotrimer, things like this. An example of this is hemoglobin. It has these 4 chains. It has 2 alpha chains and 2 beta chains, at least in the adult hemoglobin. And so this would be classified as a heterotetramer. It's got different chains and it has 4 of them. When it comes to like which chains it has and how many of each chain, now we're getting into talking about stoichiometry, which deals with how many copies of one thing you have per other thing. And so, for example, in the case of hemoglobin, we would say it had like a 2-to-2 stoichiometry or a 1-to-1 stoichiometry. So those are the levels of protein structure. We've got the primary structure, which is just a sequence of amino acids. The secondary structure, which is backbone-backbone interactions. Tertiary structure, the side chains start getting involved directly. Remember, they're just indirectly contributing here. And then the quaternary structure, where you can have multiple chains be involved. And all of these are going to lead to the characteristic, pretty functional things about proteins, structures. When we talk about now trying to characterize and talk about the actual, like the folded up protein, there are different terms that we can use. And often we want to talk about different parts of a protein. And so a word that we can use to talk about these different parts of proteins is domain. So they're kind of like rooms in an apartment. So they can be distinct rooms, which would be like a structural domain, kind of like a wall separated room. So if you have an apartment where maybe your bathroom is separated from your other rooms, like if you have a studio apartment or efficiency apartment or something like that, where you have a separate bathroom but everything else is kind of like in the same room, the bathroom would be like a structural domain. It's a distinct part of the protein. And typically when we talk about structural domains of the protein, it's something that could kind of like fold independently of the other parts. Now, this may or may not also be a functional domain, which is a place in your house where you do specific things. So this would be like the different areas of your studio apartment. So if you have your kitchen like right next to your bed, true story, then those would be like functional domains. So in a protein, you might have one domain act as like a kinase, so a phosphate adder, another domain act as a phosphatase, a phosphate remover, and things like this. And so much more on this in my post on protein domains. But these domains are often conserved between related proteins. So you can have like a kinase domain in multiple different proteins, a Walker A and B ATP binding motifs in multiple proteins, and things like this. And so domains are typically largest regions of a protein that can serve a special function or be structurally independent. So they can like fold on their own. And so it's helpful when you have these separate domains, scientists often like can separate them and study them individually, like make these individual parts by themselves and then study what they do. And much more on that again in my domain post. But I just wanted to mention too that a domain is typically like this larger thing that can then contain those motifs, which are like the furniture in the room. And so these are things like the zinc finger and things like this. These may or may not have a known function, but they're evolutionarily conserved and they're shared by many proteins. Okay, so yeah, so you have domains that can contain motifs, and these domains can be structural and or functional. Finally, let's talk about when you often see this domain like architecture laid out in figures like this, where you have these different domains in this like linear form, where these different domains are represented by these kind of like different colored regions. And often in between those, you'll see regions that don't have a distinct domain, just like kind of like lines. These lines are often intrinsically disordered regions. So intrinsically disordered regions are parts of proteins that don't have like set structures. They don't have defined structures or like permanent structures, but they're great for scaffolding. So bringing multiple things together, because if you kind of have this long loopy thing or this long kind of flexible thing, you're able to kind of lasso together different proteins. Sometimes when they bind to different proteins, and it doesn't have to be proteins, they can bind to nucleic acids, they can bind to lipids, all sorts of different things. But sometimes they adopt like a set structure. Once they bind, they kind of like snap into place. Whereas others, they kind of are always more flexible. So some proteins just have some small IDRs, so you would just see like little regions in between these domains that are more flexible. But sometimes proteins are like almost entirely IDR. And these proteins can do important things like help drive phase transition. So when you think about like things forming kind of like gels inside of cells, they can kind of help bring things together and keep them from floating away from one another. So IDRs can be really, really important, but they're also hard to study because you can't visualize them with those nice structural biology techniques that we talk about, like X-ray crystallography, which requires your protein to stand super duper still. And speaking of keeping still, although techniques like X-ray crystallography can give us sort of snapshots of what these proteins look like, proteins are constantly moving around and they can be changing their shape. And so when we talk about proteins undergoing shape shifts, we call these conformational changes. So although proteins have often, especially with those domains, they have these like strong structures that are kind of permanent. They're not totally permanent, and you can get, especially in the like linker regions between the different domains, you can get a lot of movement. So proteins can undergo conformational changes, these shape shifts, which can be subtle or dramatic. Sometimes they're induced by binding to something, or by like the pH or the other conditions inside or outside the cell or whatever. They can also be influenced by post-translational changes, so changes like phosphorylation and other changes that modify the amino acids after they're incorporated into the protein. So it's like you packed up your suitcase and now you add something new to the suitcase. You add something big and bulky to your suitcase. You add a negatively charged phosphate group to a protein. Things might have to shift around a little. So proteins are able to undergo these conformational changes because the secondary structure, where you have these backbone interactions that are really, really strong. This can be often don't get the changes within those structures, but you get them more happening in the linker regions between those domains. Although you can also have it happen inside of like the structural motifs as well. So basically proteins, although we talk about them having these distinct structures, these structures are also fluid, and they can be changing, sometimes dramatically, often more subtly. And this allows proteins to kind of serve their functions rather than just acting like bricks. Bottom line, the functions that those proteins can have is going to depend on their structure, and that structure is going to depend first and foremost on their sequence of amino acids. The sequence of amino acids is going to influence how what angles the backbone can take. When those backbone takes on that angles, it's going to leave a pattern of hydrogen bond donors and hydrogen bond acceptors coming from the backbone of the amino acids. So you have the amino acid, in the peptide bond, you have the hydrogen on the nitrogen able to act as a donor. The lone pair in the oxygen is able to act as an acceptor. You get these characteristic patterns: donor, acceptor, donor, acceptor, donor, acceptor. Then this leads to the hydrogen bonding interactions between the backbones, which is going to give you the secondary structure, things like alpha helices and beta strands, which can team up into beta pleated sheets. This is involving just the backbone of these peptides, at least directly. But now you're going to level things up to the tertiary structure. Here, the unique side chains actually get involved. You get things like dipole-dipole interactions. You get ionic bonds or salt bridges between the positive and the negatively charged amino acids. But those are actually a small part to play in the folding of a protein. The folding is primarily driven by those hydrophobic amino acids, by the exclusion of those amino acids by water, and they're kind of being forced together in the center of a protein where you can get those nonpolar interactions, those van der Waals forces and things like this. Now, all of that was involving just a single chain of the amino acid. Oh, and you can also get with cysteine bonds, you can get disulfide crosslinks, which are the only like covalent bonds that we're going to see in the tertiary structure. although these bonds are able to be broken up, unlike these like peptide bonds and things like this, the cysteine bonds are going to be more reversible. And we call the making of those bonds oxidation and their breaking reduction. Typically in a protein that is going to be intracellular, you're going to see the protein in its reduced form. So, cysteines will be individual, they're not linked together. But proteins that are secreted are often involve these cysteine cross-link. So in their oxidized form, outside the cell, it's a more oxidizing environment, and much more on the semi-post with cysteine. But most of the interactions you see with tertiary structure are just attractions and not actual bonds. And then finally, with the quaternary structure, not all proteins have this, but some do, and this involves multiple protein chains joining together and interacting with one another. So in addition, and I should also mention that in addition to having interactions between different side chains, you can have interactions between the side chains and the backbone. So there's a lot of different binding attraction opportunities, lots of different structural opportunities. There are some core structural motifs that we can talk about, like strands and helices, as well as super secondary motifs, things like zinc fingers. But all these different, and some of these domains and motifs are going to be shared between different proteins, but the proteins are each unique because they're going to have different combinations of these motifs, of these domains, of these amino acids. And so all of these proteins are going to be unique, and all of them are going to be uniquely awesome. And if scientists who study this relationship between the form and the structure, we call this structural biology. So I did my PhD training in structural biology lab, and it can sometimes be hard to get people excited about structural biology, but it's really, really cool. And so hopefully this helps you understand why the structure of a protein really is important, where it comes from, and why it matters. So yeah, so check out structural biology. It's cool. Okay, bye.